Hello and welcome to Lesson 3, Chapter 3, where we discuss the cells, which are the living units. Cells are the structural units of all living things. The human body has anywhere between 50 to 100 trillion of these tiny building blocks. Concepts collectively known as the cell theory state that the cell is the smallest unit of life. When you define the properties of cells, you define the properties of life. All, organ- all organisms are made of one or more cells. According to the principle of complementarity of structure and function, the activities of cells are dictated by their shapes and by the types and relative numbers of the subcellular structures they contain. The cell theory continues to state that cells only arise from other cells. Most body cells arise by mitosis. The trillions of cells in the human body include over 250 different cell types that vary greatly in shape, size, and function. So you can see here that we can see cells with the unaided eye. We can see cells because they're so small with the light microscope, and also using an electron microscope to see their microscopic organelles found within the cytoplasm of the cell. We have different cell shapes and sizes. Here we have cells that connect body parts, form linings, or transport gases. You see our fibroblast cells, you see epithelial cells, you see erythrocytes, erythro standing for red, cytes meaning cells, so these are our red blood cells. You have cells that move organs and body parts, that will be our skeletal muscle cells or our smooth muscle cells. You have cells that store nutrients, that will be our adipose, known as our fat cells. You have cells that fight disease, which will be our macrophages and carry out the function of phagocytosis. You have cells that gather information and control body functions, that will be our nerve cells. And then you have cells for reproduction. This will be our sperm cell that's only found in the male reproductive system, and it is the only cell of the human body that has this structure here, which is a flagellum. So this is the structure of a generalized cell. No cell is exactly like this one, but this composite illustrates features common to many human cells. Regardless of the differences, all cells have the same basic part and some common functions. The human cell has three main parts. It has the plasma membrane, the outer boundary of the cell, which acts as a selectively permeable membrane barrier. It has the cytoplasma. The cytoplasma is going to be the intracellular fluid packed with organelles, small structures that perform specific cell functions. And lastly, we have the nucleus. The nucleus, an organelle that controls cellular activities, and typically the nucleus lies near the cell's center. So from the nuclear envelope of the nucleus all the way to the plasma membrane, the barrier of the cell, the space in between is known as the cytoplasma, and it has fluid called the cytosol. Extracellular materials are substances contributing to body mass that are found outside of the cells. The major classes of extracellular materials are extracellular fluid, cellular secretions, and the extracellular matrix. So here we are with the plasma membrane. Here's the nucleus, and all of this space on the inside where the organelles are floating is the cytoplasma. What's highlighted in green will be your plasma membrane. The flexible structure separates two of the body's major fluid compartments, the intracellular fluid, which is inside the cell, and the extracellular fluid outside of the cell. The fluid mosaic model, oh, I'm sorry, this is the nucleus in green. And the plasma membrane is made up of fluid mosaic model. It depicts the plasma membrane as an exceedingly thin structure composed of a double layer or bilayer of lipid molecules with protein molecules plugged into or dispersed in it. The proteins, many of which float in the fluid lipid bilayer, form a constantly changing mosaic pattern. So here you can see membrane lipids, membrane proteins, and membrane carbohydrates. These are the components of a plasma membrane. This represents our phospholipid bilayer. So we have polar hydrophilic heads, and they are attracted to water. They lie on both the inner and the outer surfaces of the plasma membrane. And then you can see the nonpolar tails. They are going to be hydrophobic, meaning that they avoid water and line up in the center of the membrane. You can also see that phospholipid bilayer there as well. Also, a part of the plasma membrane will be cholesterol. Cholesterol makes up 20% of the membrane lipid, and its function is to stabilize the membrane, to stabilize and stiffen the membrane. A cell's plasma membrane bristles with proteins that allow it to communicate with its environment. The proteins determine what functions the membrane can perform. Many roles include transport, communication, where they act as receptors for signal molecules, and proteins can also join cells to each other and to the extracellular matrix. Proteins with different shapes will have different functions. So what you see in green are membrane proteins, the different types of membrane proteins that are found within the plasma membrane. Let's specify our different types of proteins. We have integral proteins and peripheral proteins. What you see here are our transmembrane proteins. The transmembrane proteins are going to be involved in transport, forming channels and pores. They will act as carriers. They can act as receptors for hormones or other chemical messengers and relay messages to the cell's interior. What you see here, What you see here is the pore, the channel pore found within this transmembrane protein. And here they are acting as the channel that will allow something to enter and exit the cell. Here we have our peripheral proteins, and our peripheral proteins are not embedded in the lipid bilayer like our last transmembrane protein, which was an example of an integral protein, integral meaning within. Our peripheral proteins are either found on either side of the plasma membrane. They are attached loosely to our integral proteins. You can see that it's attached loosely here to this integral transmembrane protein. They have a hydrophobic region that anchors them onto the membrane, and they act as enzymes, motor proteins, or linkers. They're going to link the cells together. Okay. There's three types of passive transport across the plasma membrane: simple diffusion, facilitated diffusion, and osmosis. All of these different types of diffusion, all of these are various types of diffusion, and diffusion can be defined as the movement of molecules or ions from an area where they are high in concentration to an area where they are low in concentration. Movement from a high to a low concentration is called concentration gradient that's moving down. It is also called movement down or along a concentration gradient. The overall effect of this movement is to scatter or disperse the particles throughout the environment. So here's a perfect example of diffusion. And here we have a dropper of blood. This dropper of blood is going to disperse the solutes into this solution of fluid, which is water. And here you're going to see random motion based on kinetic energy will make the blood move and collide constantly. And as a result, they're moving down a concentration gradient from a high concentration that was in the dropper to a low concentration. There was no blood originally in this container. So from a high concentration, a low concentration. So that's down its concentration gradient. That is diffusion. It is an example of passive transport. That blood is going to constantly move until it reaches equilibrium, okay, where it is evenly distributed. There is no more net movement of blood because there is no more concentration of it. Okay. The speed of diffusion is influenced by three factors. Concentration, the greater the difference in concentration of the diffused solute molecules or ions between the two areas, the more collision occur and the faster the particles will diffuse. The second thing that can influence diffusion will be molecular size. Smaller molecules diffuse more rapidly. The third influencing factor will include temperature. The higher the temperature, meaning more kinetic energy, it will increase the speed of molecular movement and means more rapid diffusion. In a closed container, diffusion eventually produces a uniform mixture of molecules. In other words, the system reaches equilibrium as in our third diagram here. With molecules moving equally in all directions, there's no net movement. The plasma membrane is a physical barrier to diffusion because of its hydrophobic core. Okay? Because of its hydrophobic core, here's the lipid bilayer, that two layers of those phospholipids. Here's the head, and here are the tails, the hydrophilic heads and the hydrophilic tails. Okay, so the plasma membrane is a physical barrier to diffusion because of its hydrophobic core. That is, the membrane is selectively or differentially or permeable barrier. It allows some substances to pass while excluding others. For example, it allows nutrients to enter the cell but keeps many undesirable substances out. At the same time, it keeps valuable cell proteins and other necessary substances in the cell, but allows waste to exit. Okay. So here we see this is simple diffusion. This is an example of simple diffusion. Simple diffusion is where substances diffuse directly through the lipid bilayer. Such substances are usually small nonpolar molecules that readily dissolve in lipids. These include gases such as oxygen and carbon dioxide. Other examples can include steroid hormones and fatty acids. Remember, we said that diffusion was an example of passive transport. And passive transport doesn't utilize energy, doesn't utilize ATP, because it moves down its concentration gradient. That is from a high concentration to a low concentration. So here's our plasma membrane. The lipid bilayer is going to represent our plasma membrane. This is the intracellular part of the diagram, intracellular because there's the cytosol, the fluid that's found in the cytoplasm. This is the extracellular fluid, the extracellular environment. This is found outside of the cell. Outside of the cell, we can see that we have many oxygen particles, 1, 2, 3, 4, 5, 6, 7, 8, 9, and so on and so on. But here inside the cell, you can see that there's only 5 molecules of oxygen, 5 ions of oxygen molecules. So in order for simple diffusion to take place, we're going to move down the concentration gradient from a high concentration of oxygen molecules to a low concentration of oxygen molecules. Therefore, they are going to diffuse into the cell, which is opposite of carbon dioxide. Inside the cell, we have many molecules of carbon dioxide in compared to the extracellular fluid. So carbon dioxide is going to diffuse out of the cell through the plasma membrane from a high concentration to a low concentration. This is representing simple diffusion, the movement of solutes across a plasma membrane down their concentration gradient. Here we have an example of facilitated diffusion. And the example of the facilitated diffusion will be channel-mediated diffusion. Here's where we're going to utilize channels as transmembrane proteins that transport substances, usually ions or water. You can see sodium ions and potassium ions are going to be transported into and out of the cell via this channel. Our channels can either be leakage channels or gated channels. Leakage channels are always open, and therefore they simply allow their ions or water to move according to their concentration gradients. Our gated channels are controlled, opening or closing, usually by chemical or electrical signals. And this is just another example of diffusion until we reach equilibrium. So you can see over here, water, these molecules here are going to have a higher concentration inside of the cell as opposed to outside of the cell. We only have a few water molecules, so water moves out of the cell moves out of the cell into the extracellular environment. You can see that it's done freely through the lipid bilayer, but it's also using the transmembrane protein. Okay. This slide represents tonicity. Tonicity is where many solutes, particularly intracellular proteins and selected ions, cannot diffuse through the plasma membrane. And consequently, any change in their concentration will alter the water concentration on the two sides of the membrane and results in a net loss or gain of water by the cell. Tonicity refers to the ability of a solution to change the shape of a cell by altering the cell's internal water volume. We have three types of tonicity solutions. We have isotonic solution, hypotonic solution, and hypertonic solution. Isotonic, iso meaning the same, solutions have the same concentrations of non-penetrating solutes as those found in cells. An example of this from the clinical setting will be our normal saline, 0.9% normal saline that we typically hang for individuals who are fluid depleted. Cells exposed to isotonic solutions retain their normal shape and exhibit no net loss or gain of water. So there's no net movement of water because the concentration outside of the cell is the same concentration on the inside of the cell. In a hypotonic solution, the solution is more dilute, meaning that it has more water molecules outside of the cell than inside the cell. It contains a lower concentration of non-penetrating solutes on the outside. Okay. Cells immersed in cells placed in a hypotonic solution plump up rapidly as water rushes into them. Remember, water is going to move from a high concentration to a low concentration, which is inside of the cell. And as a result of that, the cell is going to swell, burst, and eventually lyse, or what we call lysis. In a hypotonic solution, the solution has a higher concentration of solutes, so you can see that there's more purple dots in the solution as opposed to inside of the cell. And as a result of that, cells immersed in a hypertonic solution will lose water. The water is going to leave the cell to the extracellular environment, and as a result of that, the cell is going to shrivel up and go through a process called crenation. Okay. All right. So now let's talk about the different organelles that are found within the cytoplasm of the cell. Begin with our rough endoplasmic reticulum. The endoplasmic reticulum is an extensive system of interconnected tubes and parallel sacs called cisterns. The endoplasmic reticulum is continuous with the outer nuclear membrane and accounts for about half of the cell's membranes. There are two distinct varieties of the endoplasmic reticulum. We have our rough endoplasmic reticulum and we have our smooth endoplasmic reticulum. The rough endoplasmic reticulum is studded with ribosomes, which is why it is called rough. The function of the rough endoplasmic reticulum is to have the produced proteins be modified, sorted and packaged into vesicles and travel to the Golgi apparatus. The smooth endoplasmic reticulum that we see here in green is considered to be smooth because it's absent of ribosomes. Okay, it is continuous with the rough ER and it consists of tubules arranged in a looping network. The functions of the smooth endoplasmic reticulum is to metabolize lipids, synthesize cholesterol and phospholipids, and synthesize the lipid components of lipoproteins in liver cells. Another function of the smooth endoplasmic reticulum is to synthesize steroid-based hormones, detoxify drugs, pesticides, and cancer-causing chemicals. It also can break down stored glycogen to form free glucose. And lastly, it can store calcium ions. The Golgi apparatus will receive the modified and sorted proteins from the rough ER. It is known as the UPS or FedEx of the cell. Its major function is to modify, concentrate, and package the proteins and lipids made at the rough endoplasmic reticulum and export them from the cell. That is why it is called the UPS or FedEx of the cell. Lysosomes are spherical membranous organelles containing activated digestive enzymes. They are large and abundant in phagocytes such as our macrophages. They digest almost all kinds of biological molecules. They serve as the demolition crew of the cell. They digest particles taken in by endocytosis. They degrade stressed or dead cells and worn out or non-functional organelles. They perform metabolic functions such as glycogen breakdown and release. And lastly, they can break down bone to release calcium into the blood. Peroxisomes, resembling smaller lysosomes, are spherical membranous sacs containing a variety of powerful oxidase enzymes. They are going to detoxify toxic or harmful substances, including alcohol and formaldehyde. They neutralize free radicals that are damaging to our cells. And they also play a role in energy metabolism by breaking down and synthesizing fatty acids. The mitochondria is known as the powerhouse of the cell, providing most of the cell's ATP, adenosine triphosphate, supplying most of its energy. Mitochondria are very complex organelles. They contain their own DNA, RNA, and ribosomes and are able to reproduce. Membrane-bound ribosomes. So ribosomes are small, dark stained granules composed of proteins and a variety of RNAs called ribosomal RNAs. Ribosomes are going to function as the site of protein synthesis. We have membrane-bound ribosomes and we have free ribosomes that are floating in the cytoplasm. The membrane-bound ribosomes are going to be bound to our rough endoplasmic reticulum. Our free ribosomes are going to float freely in the cytoplasm, the little green structures that you see in the cytoplasm. Both, whether they are bound or freely floating, are going to be the site of protein synthesis. The cytoskeleton, literally the cell skeleton, is an elaborate network of rods running through the cytosol and hundreds of accessory proteins that link these rods to other cell structures. Our cytoskeleton supports our cellular structures and provide the machinery to generate various cell movements. There are three types of rods in our cytoskeleton. They include our microfilaments, intermediate filaments, and microtubules. Let's discuss our microfilaments. They are the thinnest. They are going to be made up of the protein actin. Their function is cell motility and cell shape. They are going to constantly break down and reform smaller subunits whenever and wherever their services are needed. Our intermediate filaments are tough, made up of insoluble protein fibers. They are the most stable and permanent of the cytoskeleton elements, and they strongly resist tension. They attach to our desmosomes, and their main job is to act as internal cables to resist pulling forces exerted on our cells. Lastly, the microtubules are going to have the largest diameter. They are hollow tubes made up of spherical protein subunits called tubulin. They are remarkably dynamic organelles constantly growing out from an area called the centrosome. They determine the overall shape of the cell. They are anchored at one end in a region near the nucleus called the centrosome or the center. This is where our microtubules are found in the centrosome or the cell center. The centrosome acts as a microtubule organizing center. It is also going to have centrioles, which you see highlighted in green in the centrosome region. The centrioles are small barrel-shaped organelles oriented at right angles to each other, and they are best known for generating the mitotic spindle that takes place or that is developed during cell division. Centrioles also form the basis of cilia and flagellum. Cilia are whip-like motile cellular extensions of the plasma membrane that occur typically in large numbers, and they're going to move substances in one direction. Microvilli are tiny finger-like extensions also of our plasma membrane. Here's the cilia and here are the microvilli. So they are smaller, tinier than our cilia. They are also extensions of the plasma membrane of the cell. They're going to function to increase the plasma membrane surface area. They are typically found on the surface of absorptive cells, such as our intestinal and kidney tubule cells. Tight junctions, desmosomes, and gap junctions are part of our plasma membrane. Our tight junctions help prevent molecules from passing through the extracellular space between adjacent cells and restrict the movements of membrane proteins. They are called tight junctions because they prevent things from entering and exiting the cell. Our desmosomes serve as anchoring junctions on the cell, They bind neighboring cells together into sheets and also contribute to a continuous internal network of strong fibers that act as guide wires or glue of cells. And as cell membrane junctions will be the gap junctions. And our gap junctions is a communicating junction between adjacent cells. Our gap junctions are going to allow things to pass from one cell into a neighboring cell. Certain things that can pass through will be ions, sugars, and small molecules. Substances move through the plasma membrane in essentially two ways, by passive transport or active transport. Passive transport, which was our simple diffusion, facilitated diffusion, and osmosis, they cross the membrane without energy input from the cell. Therefore, they do not require ATP. Our active processes, the cell provides the metabolic energy, ATP, needed to move substances across the membrane. Anything that works best when it is controlled. For our cells, the control center is the genetic material containing nucleus. As the genetic library, it contains our genetic material, the instructions needed to build nearly all of the body's proteins. Most cells have only one nucleus, but some, including our skeletal muscle cells, our bone cells, and some liver cells, are considered multinucleate, meaning that they have many nuclei. An exception to this will be our mature red blood cells, where their nuclei is ejected before the cells enter the bloodstream. All of our body cells have nuclei except for our red blood cells, our erythrocytes. So they are considered anucleate, without a nucleus. Therefore, they cannot reproduce, and they live in the bloodstream for only 3 to 4 months before they deteriorate and are destroyed in the spleen by our lysosomes. Without a nucleus, our red blood cells cannot produce messenger RNA to make proteins, and when its enzymes and cell structures start to break down and become destroyed, they cannot be replaced. The nucleus has 3 recognizable regions or structures. It has the nuclear envelope, and it also has a nucleolus, and also it has chromatin. Here's the nucleolus. The nucleolus, or also called nucleoli, is a dark stained spherical body where the ribosomal subunits are assembled. The function of the nucleolus is to produce our ribosomes. The nuclear envelope surrounds the nucleus. It has pores, openings in it. These pores regulate entry and exit of molecules into the nucleus. The nuclear envelope is also has selective permeable characteristics just like the plasma membrane. Seen through a light microscope, chromatin appears as a fine, unevenly stained network, but special techniques reveals it as a system of bumpy threads weaving through the nucleoplasma. Chromatin is composed of 30% DNA, which is seen here, which is our genetic material. 60% is made up of histones, which package and regulate the DNA, and 10% RNA chain, newly formed or forming. The fundamental units of chromatin are nucleosomes, which consist of flattened, disc-shaped cores of 8 histone proteins connected like beads on a string by a DNA molecule. The DNA winds twice around each nucleosome and continues on to the next cluster of histones. Histones provide a physical means for packing the very long DNA molecules in a compact, orderly way, but they also play an important role in gene regulation. When a cell is preparing to divide, the chromatin threads coil and condense enormously to form short, bar-like bodies called chromosomes, which you see here. Here represents the genetic code, the rules by which the base sequence of a gene is translated into an amino acid sequence called genetic code. You could see each code is made up of a 3-base sequence, and this is found on the DNA. A 3-base sequence found on a DNA, and it's known as a triplet. So here's the 3-base sequence, and it is known as a triplet. The corresponding 3-base sequence on the messenger RNA is called a codon. A codon. Because there are only about 20 amino acids, some are specified by more than one codon. Each codon specifies for a specific amino acid. The start codon is represented by the triplet AUG, and it is going to code for the amino acid methionine. That the genetic information of a cell is translated into the production of proteins via a sequence of information that is completely determined by complementary base pairing. The transfer of information goes from DNA-based sequence triplets to the complementary base sequence of messenger RNA codons, and then on to the transfer RNA-based anti-codons. In addition to the nucleus serving as the control center of the cell, it also includes our genetic material. The genetic material is going to consist of DNA and RNA. Cellular activities are dependent upon our DNA for production of our proteins. This introduces the concept of central dogma. DNA is going to be transcribed into RNA. RNA is going to be translated into protein. So DNA serves as the blueprint for protein synthesis. In addition to directing its own replication, DNA serves as the master blueprint for protein synthesis. We define a gene as a segment of DNA molecule that carries instructions for creating a polypeptide chain, which will result in the synthesis of a protein. 4 nucleotide bases, adenine for A, guanine for G, thymine for T, and C for cytosine, are the letters of the genetic alphabet. The information of DNA is found in the sequence of these bases. Each sequence of 3 bases is called a triplet, as I mentioned before, and can be thought of as a word that specifies a particular amino acid. Variations in the arrangement of adenine, thymine, cytosine, and guanine will allow for our cells to make all the different kinds of proteins needed. Transcription transfers information from a DNA-based sequence to the complementary base sequence of a messenger RNA molecule. The form is different, but the same information is being conveyed. Once the messenger RNA molecule is made, it detaches and leaves the nucleus via a nuclear pore and heads for the protein synthesis machinery, which is the ribosome. Translation. A translator takes a message in one language and restates it in another. In the translation step of central dogma or protein synthesis, the language of nucleic acids, which are the base sequences, is translated into the language of proteins, which is the amino acid sequence. Both transcription and translation involves 3 basic phases. Initiation, elongation, and termination. There are DNA can never leave the nucleus, so DNA requires not only a decoder but a messenger as well. The decoding and messenger functions are carried out by RNA, the second type of nucleic acid. RNA differs from DNA in that RNA is single stranded, whereas DNA is double stranded. RNA has the sugar ribose instead of deoxyribose for DNA, and the base uracil, U, is going to be the nitrogenous base found in RNA instead of T, the thymine. Three forms of RNA typically act together to carry out DNA's instructions for polypeptide or protein synthesis. Starting with our messenger RNA, it carries the coded information to the cytoplasm where protein synthesis occurs. It is the messenger. Our transfer RNA is smaller than all the other types of RNA, and it's going to transfer or ferry the amino acids to the ribosomes, where they're going to decode the messenger's RNA message for amino acid sequence in the polypeptide or protein to be built. Ribosomal RNA, along with proteins, form the ribosomes, which consist of two subunits, one large and one small, that serve as the sites of protein synthesis. Here are the three stages that are found in both transcription and translation: initiation, elongation, and termination. Here you can see the nitrogenous bases. We have thymine, adenine, cytosine, and guanine for our DNA. And for RNA, the bases are adenine, uracil, guanine, and cytosine. Guanine always binds with cytosine. Adenine always binds with thymine. Remember, thymine is not found in RNA, so adenine will also bind with uracil. This represents our transfer RNA, where it's going to transfer that codon and meet up with the anticodon in the newly transferred polypeptide. This is the small subunit and the large subunit of the ribosome. The cell cycle is a series of changes in a cell can go through from the time it is formed until it reproduces. Interphase, seen in the green, is where the cell grows and carries on its usual activities. Cell division, or the mitotic phase, seen in yellow, is where it divides into two genetically identical cells. Interphase is the period from cell formation to cell division. It is also known as the metabolic phase or the growth phase. It has subphases. It is divided into G1, S, and G2. The G stands for gap. The S stands for synthesis. The G1 phase, the light green stage here, is metabolically active, synthesizing proteins rapidly and growing vigorously. The G1 stage typically lasts several minutes to hours, but it may last for days or even years. The S phase, or synthesis phase, is where DNA is replicated. Remember, DNA replication was the first step in the central dogma. DNA is replicated here, ensuring that the two future cells being created will receive identical copies of the genetic material. The darker green stage here is GAP2 subphase, known as G2. It is the final phase of interphase. It's brief. This is where enzymes and other proteins needed for division are synthesized and moved into their proper sites for cell division. When the cell ensures that all the DNA has been replicated and damaged DNA has been repaired, the cell is now ready to enter into the mitotic phase. The mitotic phase is known as cell division. It's essential for body growth and tissue repair, such as cells of the skin and the intestinal lining reproduce themselves almost continuously. Others, such as liver cells, divide more slowly to maintain the size of the organ that they compose, but retain the ability to reproduce quickly if the organ is damaged. Nervous tissue, skeletal muscle, and heart muscle lose their ability to divide when they are fully mature, and repairs are made with scar tissue. In most body cells, cell division, which is called the mitotic or mitosis phase, involves two distinct events: mitosis and cytokinesis. Mitosis is the division of the nucleus. It is a series of events that parcels off the replicated DNA of the parent cell to two genetically identical daughter cells. Mitosis is described as four phases: prophase, metaphase, anaphase, and telophase. Mitosis is usually a continuous process with one phase merging smoothly into the next. It typically lasts about an hour or less. Cytokinesis is the division of the cytoplasm. It begins during late anaphase and is completed after mitosis ends. It draws the plasma membrane inward to form a cleavage furrow over the center of the cell. This furrow will continue to deepen until it pinches the cytoplasmic mass into two parts, yielding two daughter cells. The daughter cells will then enter the interphase portion of the cell cycle and wait its turn to divide. This cell cycle continues over and over and over again. So let's begin the cell cycle with interphase. Okay? Interphase is the period of a cell's life cycle when it carries out its normal metabolic activities and growth. It is the longest stage of the cell cycle. During interphase, the DNA containing material is in the form of chromatin. You can see the chromatin here. The nuclear envelope is easily visible, and the nucleolus or nuclei is also visible and intact. Let's start the mitosis phase of the cell cycle, and it begins with prophase. In prophase, the chromatin coils and condenses, forming chromosomes. The nucleoli disappears. The nuclear envelope starts to break up. And in the centrosomes, the centrioles start forming the mitotic spindle. Metaphase is the second stage of mitosis. Chromosomes are going to align in the middle of the cell along the equator or the metaphase plate. Anaphase starts as the spindle fibers move the sister chromatids apart to opposite poles of the cell. Anaphase are when the sister chromatids that make up the chromosome split apart and move to opposite poles of the cell. It is the shortest phase of mitosis. And telophase is the final phase in mitosis. It is prophase in reverse, meaning now that the chromosomes are going to uncoil and resume thread-like chromatin form, a nuclear envelope forms, the nucleolus reappears, and the cleavage furrow continues to develop. This stage will continue as cytokinesis as the cleavage furrow deepens. Cytokinesis is known as cytoplasmic division, and we are going to end with two genetically identical daughter cells. So here we can see our cell cycle. interphase, which is the longest stage of the cell cycle, mitosis, and ends in cytokinesis. Thank you.